we get into things, you will notice that your mics and your cameras are all turned off. That is to not allow any disturbances throughout the entire session. Also, guys, we please ask you guys to state your name, your surname, and your school's name in the comment section. And also the number of people that is viewing from your device. So if you are viewing from your school, you can just state your teacher's name as well as your school's name, as well as the number of people viewing from your teacher's device. If you guys have any questions, you guys can just pop that in the chat, and myself and my colleague will get to that as soon as possible. Okay, so let's get into things, guys. Hope you guys are ready for it. Okay, so we are quickly just going to go through the format of the paper. So paper 2 is made up, is 2.5 hours and is made up of 115 marks, and it's split into section A and B. Section A being your shorter questions, so your multiple choice, your terminology, where they give a statement, and then you have to figure out what that statement is referring to, matching columns, that makes up 50 marks. And your longer type questions is equal to 100 marks, and yeah, that will mostly be about either graphs or diagrams that you need to interpret. Okay, so in paper 2, you cover 4 main topics. The first one is DNA code of life, which makes up 27 marks. Meiosis making up 21 marks. Then the bulk of your paper being genetics and inheritance, which make up 48 marks. And evolution, which makes up 54 marks. So those 2 topics are the bulk of your paper. Okay, so a few things just to take note of. So you guys will see here are just some tips for you guys. So it's quite important to look for question words when you're answering questions. This word will tell you what to do and it will help you to understand what the examiner is asking of you. So just please take a picture of these words that's listed over here. Just make sure you understand what the words mean. So, for example, when they say compare, they are referring to you need to compare similarities and differences between, for example, if talking about evolution, you need to compare differences and similarities between, let's say, Homo sapiens and another type of species like Australopithecus. So just take note of that. And yeah, I'm not going to go through all of this, but you guys can just have a look, read, and also when you're working through question papers before your exam, just take note of what question words are asking and just try and understand when you're looking at the memo how they're answering those question words. Okay, so that's it for that, guys. So let's move on to our first topic. So I'm just going to cover a little bit of theory, right? So let's quickly look at a bit of DNA replication. Okay, so what is DNA replication? So it's the duplication of the DNA molecule to form 2 identical copies, right? So replication generally occurs during the interphase of the cell cycle and occurs in the nucleus of the cell. So again, just take note of where the replication takes place within the cell, and they like to ask that for about 1 or 2 marks. And then also when they say, they also might ask you what just define DNA replication. It's basically, as I said, this first part over here, the duplication of the DNA molecule to form 2 identical copies. Okay, next part. So functions of DNA. Again, this type of question generally goes about 2 marks. So what is the function of DNA? DNA makes up genes which carry hereditary information, right? That's 1 mark. Second mark would be DNA code contains coded information for protein synthesis. Okay. Then, what is the importance of DNA replication? So this again goes for another 2 marks, 1 or 2 marks. So again, also when they ask these questions, there's 1 or 2 more questions, try and be specific and concise and to the point. Don't, in like the beginning, start like having like an introduction type of thing. Just go straight into the point, because when they mark, they will, whatever's written first in your first 2 lines, they're going to mark that. And if your answer's in the 3rd or 4th line, they're not going to continue marking, then you lose the marks. So just be very concise when you're answering your questions. So what is the importance of DNA replication? So during mitotic cell division, 1 mother cell divides into 2 daughter cells. So if this is a 2 mark question, that in the bold over here, 1 mother cell divides into 2 daughter cells, that is your first mark. Your second mark would be each daughter cell has identical DNA composition to the mother cell, because remember, DNA duplication is a duplication of the DNA molecule to form two identical copies. So in the bold over here, identical DNA composition to the mother cell, that would be your second mark. Okay, so let's move on to the explanation of what is DNA replication. Let's look at the process and let's just see where we get our marks. Okay, so with this one, this question is about 6 to 7 marks generally. If it is less marks, we'll get to that in a bit. Okay, so DNA replication. So what is it? Firstly, you have the double helix, which unwinds over here. And then you have your weak hydrogen bonds between the nitrogenous bases. So they break and the two DNA strands, they separate or they unzip as seen over here. Next up, what you have is both the DNA strands now, the original strands will now serve as templates on which you build your new DNA molecule. Then you have free nucleotides in the nucleoplasm are used to build a DNA strand onto each of the two original DNA strands by attaching to their complementary nitrogen spaces. So as you guys know, adenine binds to thymine and cytosine binds to guanine. Okay, then next up, this will then result in two identical DNA molecules, and each molecule consists of one original strand and one new strand. Okay, so that would be about 6 or 7 marks. Okay, so now let's look at a question that has to do with DNA replication. Okay. So this is from a question 2 from Eastern Cape, September 2021. So question 2 reads, the diagram below shows two forms of a chromosome, right? So in diagram 1, this is an unreplicated chromosome. In diagram 2, we have a replicated chromosome, right? So let's look at what they ask. So they say identify part A. So part A, let's quickly go back to the other picture. It's a bit better. So part A, they point to the black circle over there, right? Okay. So what is part A? Part A is the centromere, right? So I know people might get confused what is centromere, and there's also another one called centrosome. So the centromere is the site where the two chromatids are basically joined together, right, in a replicated chromosome, and where the chromosome is then attached to the spindle thread or spindle fibers during cell division. Now, the other one that has a similar name, centrosome, that is where your spindle fibers are formed from. They basically form the spindle fibers. Okay. So again, they're using an our question word identify, and that means state the essential characteristic. So basically quite simple and straightforward over there. Okay, then the next question. It says identify the process that led to the formation of a chromosome represented by diagram 2. So in diagram 1, like I said, we have the unreplicated chromosome. In diagram 2, you have the replicated chromosome. So that tells us what we're looking at is DNA replication. Okay. So that's pretty straightforward and simple 1 mark answers, 1 mark questions. Okay, so moving on. Next up, they ask you describe the process mentioned in question 2.1.2. That's worth 4 marks. So again, like when they say the word describe, they want you to state in words the main points of the structure, right? So again, let's go through it. So this is a 4 mark question, but remember when I went through DNA replication in the previous one, that is about 6 or 7 marks. But you can list the entire process and then they will just mark everything basically till like the 4th point that you made, as long as everything is in chronological order. Okay. So let's go through the explanation again of DNA replication. So the DNA molecule, it unwinds and unzips, as we said. This answers more condensed version of what we went through earlier. Then next up, we have the weak hydrogen bonds that break, and each strand serves as a template the free-floating nucleotides in the nucleoplasm join to their complementary strand or base, where thymine binds to adenine and guanine binds to cytosine, forming two identical strands of DNA. Right, so that would basically be your 4 marks, but like I said, you can list everything and they'll just mark the first 4 points. Okay, so for 2 marks, they say give 2 reasons, only 2 reasons, why the process described in question 2.1.3 is important. So they want to look at the importance of DNA replication, right? So we went through this. So firstly, produce 2 exact copies of the chromosome so that each cell formed in mitosis has the exact genetic material or same amount of DNA as the parent cell. So again, first mark, produce 2 exact copies of the chromosome. Then your second mark comes in when you say has the exact genetic material or same amount of DNA as the parent cell or chromosome. So that is where you get your 2 marks. Okay, so perfect, guys. That's very simple and straightforward type question. So let's look at a few more types of questions that they can ask you under the topic of DNA. Okay, so this is question 1 from KZN, September 2024. Okay, so the question reads, DNA was extracted from the cells of a cat and was analyzed for its nitrogenous base composition. It was found that 26% of the bases are guanine, right? Okay, so some important information to take note of there. 26% of the bases are guanine. So now they want to know what percentage of the bases are thymine. So they give you a few options. So this basically the information that we're going to work with is this over here. So let's look at it. So from your studying and your knowledge, in DNA, the amount of guanine is always equal to the amount of cytosine, and the amount of adenine is always equal to the amount of thymine, and that is based on Jar-Graf's rule, right? And they're equal amounts of guanine and cytosine, and they're equal to each other, and the amount of adenine to thymine is also equal to each other. That is crucial for the structure of DNA to ensure that the bases are properly paired together, and because the bases are properly paired together, that basically ensures the proper structure of the double helix. So I don't have to get into that too much. Just wanted to explain a bit of that to you guys. Okay, so let's just quickly do a bit of calculations over here. So if we have 26% of guanine, that means because guanine and cytosine are in equal amounts in DNA, so because we have 26% guanine, we will then have 26% of cytosine, right? So in total, that is going to be 52%, right? So the remaining percentage of bases will be the combination of adenine and thymine. So in DNA, the 4 nitrogenous bases, they make up 100% of the DNA base composition, right? So what we're going to say is simple. You're just going to say 100% minus 52. That's going to give you 48%. So now 48% is basically the combined amount of adenine and thymine, right? Okay. So simply to get the amount of the percentage of the bases that are thymine, you're basically just going to take the 48, you're going to divide it by 2, and you're going to get 24%. So 24% of the bases are then thymine. And then the other 24% would be adenine. So if you take that 48 plus 52 gives you your 100%. Because like we said, the 4 nitrogenous bases, they make up 100% of the DNA base composition. Okay, so pretty simple there, pretty straightforward. So your answer over here would be C. Cool. Okay, So let's move on to another type of question relating to DNA. So this is just another version of it. Okay, so this is from Eastern Cape, September 2024. So they ask, how many nitrogenous bases are found in a protein made up of 66 amino acids, right? So basically, guys, as you guys know from your study, each amino acid is made up of 3 nitrogenous bases, right? It's made up of a sequence of at least 3 nitrogenous bases. So very simple, very straightforward what you're going to do. You're simply going to take 66 amino acids and you're going to multiply it by 3 because 3 bases make up 1 amino acid, and that's going to give you an answer of 198. The answer over there would be A. Okay, so those are 2 ways in which they can ask you the questions based on DNA. Okay, I hope you guys understand that. Okay. So now we're going to move on to another type of question. This is a bit more of a longer type of question on DNA. So this is question 2 from November 2017. And basically what this question says is, the sequence of amino acids in a protein molecule is coded for by DNA and RNA. The table below shows some mRNA codons and the corresponding amino acids. Okay. So, first question. According to the table, how many codons code for phenylalanines? That's with 1 mark. So basically, just read from the table. So we can see over here, alanine, we got UUC is the 1 and triple U. So answer is 2. Next question. What is the anticodon for glutamic acid? Okay. So we have the codon for glutamic acid. So over here we have the codon, the codon GAG. That is the codon. So now we want to find the anticodon. So that is going to be simply CUC. Right? Because remember, you need to find the complementary base pairing. So the complement of guanine and cytosine, complement of adenine is uracil. Because remember, we're working with RNA. Anticodons refer to working with RNA. Okay. So that's also pretty simple over there. Then the next question. So it says a section of mRNA has the following sequence. Okay, so a section of mRNA has the following base sequence and is read from left to right. So what do they want to know? They say give the question A DNA base triplet for the last codon on the section of mRNA. So they want to know the DNA base triplet for the last codon on the section of mRNA. So they're referring to ACC, which is over here. So basically, as you guys know, the mRNA is derived from the DNA. And now we need to replace each RNA nitrogenous base with its DNA equivalent. So we got adenine, cytosine, and cytosine. So the DNA equivalent of adenine is not going to be uracil, because uracil is RNA. It's going to be thymine. The DNA equivalent of cytosine is going to be guanine, and same for this one over here. So your answer over here is going to be TGG, because you're replacing the RNA nitrogenous base with its DNA equivalent. Okay. There we go. Then the next question says, what is the first amino acid coded for by the section of mRNA. So quite simple. We're just going to look at GAAU. So that is the codon found over here, and the amino acid that's coded for by that mRNA codon would be aspartate. Okay, so that's pretty simple, guys. Okay, let's continue with this question. So, okay, so this says, this is basically the same question as before. A section of mRNA has the following base sequence and is read from left to right. So I'm calling that the original sequence. Let's look at the new question. A mutation occurred which resulted in the following base sequence on the mRNA molecule. So it's saying a mutation occurred which resulted in the following base sequence on the mRNA molecule. So let's look at all codons. So we have codon 1 over here is the same. Codon 2 is the same. Codon 3 is the same. But here's something different in codon 4. Codon 5 is the same. Codon 6 is the same. Okay, so now we know there is a mutation that occurred on codon 4. Okay. So let's see what they want to know. They are saying describe the mutation that occurred. So basically explain what you are seeing. So what mutation occurred? Basically on the fourth codon, cytosine at the end of the fourth codon, cytosine changed to uracil, right? So you can say C was placed with U on the fourth codon. That is one way of saying it. Because how do you get the 2 marks? You're saying what occurred, the C was placed with U. In your second mark, you're saying the location of where the mutation occurred. That's how you get you 2 marks. Or you can say AGC, the fourth codon, changed to AGU. So there's 2 ways of saying it. But remember, first mark you get by saying where the change, first mark you are explaining what the change is. Second mark is where the change occurred. Okay, so as you guys can see, you have to also look at both the current question and the previous question. Just take note of maybe where they are referring to. Sometimes they're not very clear, so just have a look and try and understand what they are referring to. Okay, so moving on to the second part of this question, it says explain the effect that the mutation described in question 2.1.4a will have on the resulting protein. So maybe there's a change, maybe there could be a completely new protein that is formed, or maybe it could be the same protein that is formed. So in order to understand what effect the mutation has, we're going to have to then refer back to our table, right? So as you guys can see, so AGC, so the codon AGC, that coded for serine, right? Now the new mRNA codon AGU, that also codes for serine. So there's no change in the protein that will be formed, right? So it goes with the same amino acid, serine. That is 1 mark. Second mark would be, therefore, there will be no effect and the same protein will be formed. Okay. So pretty simple, guys. Pretty straightforward over there. Okay. So let's move on to the next part of, or the next topic under paper 2, and that is going to be meiosis. So I'm just going to explain a bit of theory there again, and then we're going to go through a few questions. Okay, so meiosis. So what is meiosis? It's a type of cell division whereby a diploid cell, a somatic cell, undergoes 2 cell divisions and divides to form 4 haploid cells, that is your gamete or your sex cells. So meiosis takes place in the testes and ovaries of animals and in the anthers and ovules of plants. Okay, so again, definition, about 2 marks, and you're basically just going to say the first point over there. They might also ask you, where does meiosis take place? In males or females? That would be either 1 or 2 marks. So it would be your second point over here. Okay. So let's just look at a nice simple way to remember meiosis. So we got this mnemonic acronym IMPMAT. So let's just quickly go through it. So I stands for interphase, or I for in between, the part of the life cycle of the cell that is in between cell divisions. P for prophase, for P for preparation. The chromosomes prepare for meiosis by untangling and becoming clearly visible. Crossing over also takes place, but remember that only occurs in prophase 1. Then metaphase, M for the middle, so that's quite distinct to identify when is metaphase occurring. Just have to look where the chromosomes, homologous chromosomes or chromosomes are aligned at the equator, or the chromosomes move to the middle or the equator. Anaphase, A for apart. So the chromosomes or chromatids move apart or move to the poles. So that basically is when the spindle fibers are pulling them apart. So just take note of that key distinction between metaphase and anaphase. And then telophase, T for terminal, the final phase of meiosis 1 or 2. Okay, that's just if you're struggling to understand or remember the process, just look at this little trick to remember it, and just look at the keywords M for middle, A for parts, quite distinct to identify the different processes. Okay. So now we're just going to look at the differences between meiosis 1 and meiosis 2. All right. So let's look at it. So the first difference. So the chromosomes arrange at the equator of the cell in homologous pairs. So in meiosis 1, so remember the chromosomes arrange at the equator of the cell in homologous pairs. So they're referring to metaphase 1. Because now they say homologous pairs. Chromosomes line up at the equator of the cell individually, as you can see over there. So chromosomes, or they'll say chromatids. So you know they're referring to metaphase 2. Then whole chromosomes move to opposite poles of the cell. This is now referring to anaphase 1, as seen over here. And you guys will see that the spindle fiber will start to pull the chromosomes, the whole chromosomes, towards opposite poles. Then in meiosis 2, the daughter chromosomes or chromatids move to opposite poles of the cell, as indicated over here. Now, 2 cells form at the end of this division. That is telophase 1, and then 4 cells are formed at the end of this division. That is telophase 2. In meiosis 1, the chromosome number is half during meiosis 1, and the chromosome number remains the same during meiosis 2. And then also just take note in meiosis 1, crossing over only takes place in prophase 1, but no crossing over takes place in meiosis 2. Okay, so just some distinctions you guys have to take note of over there. Okay. So let's move on to questions dealing with meiosis. So looking at Gauteng 2019. So the diagram below shows different phases of meiosis in an organism. So now you just have to sort of analyze it. You can make a couple of notes on the side when you're busy with your papers. Just do some key things that you pick up. Just make notes of that. Also kind of helps you guys in the exam. Okay, so what do they want to know? So they say provide labels for A, B, and D. So A over there is pointing to the outside of the cell, so that's referring to the cell membrane. B is referring to over there, like sort of containing 2 chromosomes. So B is referring to the homologous chromosomes because they have 2 sets of chromosomes. Then what is D referring to? So D is actually referring to the spindle fibers because they're pointing at the stacked bits. So that would be the spindle fibers. Okay. So quite simple over there. You just look at the diagram and you just interpret what you are seeing. So next question. They said give 2 reasons why chromosomes B are regarded as homologous. So just to now kind of look and analyze at 2 chromosomes. So firstly, what we can say is these chromosomes are of the same size. They have the same shape as well as the same length. Okay, so you get your 2 marks for just stating those 3 points over there. Okay, next question. It says identify the phases shown in diagram 1, 2, and 3 respectively. So in diagram 1, this one over here, as you guys can see, they are at the equator. The chromosomes are at the equator, but not just chromatids. It's homologous chromosomes and it's not single or individual chromosomes, homologous chromosomes aligned at the equator. So that is then referring to metaphase 1. Then in diagram 2, it's referring to prophase 2, right? So it could either, you would think it's either prophase 1 or prophase 2, but it's actually referring to prophase 2 because if you look, so sorry, in both prophases, firstly, your nuclear membrane and your nucleolus start to disappear as indicated by the dotted line surrounding the homologous chromosomes, right? So that's the first thing. And also, we know it's not prophase 1, it's rather prophase 2 because there's no indication on the chromosomes that crossing over has taken place. Therefore, we know it's not prophase 1, but prophase 2. Okay, then identify the identify the phases shown also in diagram 3. So diagram 3, what we note is that we have single chromosomes, not now homologous chromosomes, are single chromosomes. They are basically arranging themselves at the equator. Right? So they're not being pulled to opposite poles. We can still see the spindle fibers are quite distinct over there. So it's rather metaphase 2. So again, it's metaphase 2 because your single chromosomes are lined at the equator. They're still in the middle of the cell. They're not on the ends of the cell. So that's how we know it's metaphase 2. And also the spindle fibers are quite distinct and there's no a real indication of the chromosomes being pulled to the opposite ends. Therefore, it's metaphase 2. Okay. So now they want to know, give a reason for identifying the phase in diagram 3. So we basically gave that. We have a single row of chromosomes at the equator. So just saying that single statement gives you 2 marks. Okay, perfect, guys. And then last question. Identify 2 events in meiosis that contribute to genetic variation. So we need to list 2 events. So first one, in prophase 1, you have crossing over that takes place. And then in your metaphase, you also have metaphase 1 and 2, you have the random arrangement of chromosomes along the equator. So that would be your 2 marks over there. Okay, so let's look at another question that has to do with meiosis. So this is from Eastern Cape, September 2021. So the diagram below represents a cell during cell division. So you guys can see they have labeled different parts. So again, analyze the diagram, try and understand what is happening, and then start with the questions. Okay, so first question, name the type of cell division shown in the diagram above. So name, they just basically with that question word name, they just want you to give the name of something, quite simple, quite straightforward. So the name of the type of cell division would be meiosis. That's quite simple. Obviously, you guys know the different phases, and this diagram appears to be referring to the metaphase. You guys can see the spindle fibers are also distinct there. So they're referring to meiosis as the name of the type of cell division, metaphase being the phase in meiosis. Okay. Next question. Identify the phase represented by this diagram. Metaphase 1 because homologous chromosomes are aligned at the equator. Then next, they say give the letter or letters that represents A, the structure that moves or pulls chromosomes or chromatids to the poles during cell division. So again, I should analyze the diagram and just make sure you are writing the correct letter. So for A, the structure moves or pulls chromatids to the poles during the cell division. That would be B, that is the spindle fibers that move or pull chromosomes to the opposite poles. Next question, the part that is responsible for forming the spindle fibers, that would be F, your centrosome, not centromere, because remember again, centromere holds the 2 chromatids together. And then 2 chromatids that are identical, so that would be D and E. Okay, because they're pointing at the same chromosome pair. Okay. Okay, let's look at the next set for this question. So, how many chromosomes will be found in each daughter cell at the end of the cell division? So, at the end of meiosis, how many chromosomes will be found? That would be 4. Okay. Then they want to know, give the name of the cells that will be formed as a result of this type of cell division in a male. So in a male, it would be your sperm cells, won't be a gamete. In your female, it would be the egg cell. So just take note of what they're referring to. Okay. So guys, now we're going to move on to a bit of genetics. So let's get into it. So we are looking now at Gauteng province 2023. So it's quite a bit of information here, but let's go through it. They are saying now that cats inherit many genes that affect the appearance of their coat. One gene controls whether the coat is striped or solid colored. The agouti allele, capital A for striped hair, is dominant over the allele for hair that is solid color. A second gene affects the length of the coat. Cats can either have long hair or short hair. Long hair L, capital L, is dominant over short hair, small l. And then they show you pictures over there. And then another statement they make is a male who is heterozygous for both agouti and long hair mates with a short-haired, solid-colored female. Okay, so quite a bit of information, but let's look at the first question and take it from there. So they say state the genetic cross represented above. So state basically refers to write down information about discussion. It's with 1 mark, so you don't need to justify your answer. You can just write what you think. So what type of genetic cross are we looking at? It's obviously a dihybrid cross because we have dihybrid cross, remember, is inheritance of 2 different traits, and that is each controlled by different genes. So as I state over here quite clearly, one gene controls coat color, a second gene controls the length of the coat, and that clearly indicates we're looking at a dihybrid cross. Next question, give the genotype of the female cat. So the female cat, as they state over here, so the male heterozygous and mate to the short-haired, solid color female cat. So the female cat has recessive genes. So her genotype would be the following over there. Okay. So the female is recessive for both coat color as well as short hair. Okay, next question. What is the possible gametes produced by the male cat? Okay, so the male cat, firstly, he is heterozygous for both conditions, right? Okay, so the male's genotype, so he's heterozygous for both agouti and long hair. So this would be his genotype, and obviously because he's agouti, he has the dominant allele for that, and he also has long hair and is heterozygous for that as well. So that would be the male's genotype. So now we can get the possible gametes from the genotype. So basically you look at the genotype and you look at how many possible combinations you can make from that. Okay, so let's look at the possible combinations. So basically you're kind of just combining each allele from the one trait with the allele from the second trait. Okay. So they are the possible combinations. Right. So let's move on to the next part of this question. And the question would like to know, name the inheritance shown by coat appearance traits mentioned above. So this is actually referring to complete dominance. So the type of inheritance shown by codependent traits mentioned above would be complete dominance, because complete dominance is where one allele is dominant and the other is recessive, so such that the recessive allele masks the expression of the the recessive allele is masked by the expression of the dominant allele. So, for example, the male is heterozygous for both West Coast appearance, and he had the agouti allele, and that masks the expression of the solid color allele. Okay, right. So basically, it can't be, let's say, incomplete dominance, because neither one of the two alleles of a gene is dominant over the other. So yeah, in incomplete dominance, neither one of the two alleles of a gene is dominant over the other. And in incomplete dominance, you will have an intermediate phenotype that will result in the heterozygous condition. So basically, if you had a red and a white flower crossing, they will result in a pink flower. So that would be your incomplete dominance. So that's not happening in this case. It can't also be codominance, because codominance, an example of that would be a blood type, because both alleles of that gene are equally expressed in the genotype, and it's both equally expressed in the heterozygous condition, so it can't be codominance. Okay, so remember it's complete dominance because the agouti allele completely masks expression of the solid color. Okay, so let's move on to the next question. So they want to know what percentage of the offspring will inherit the same phenotype as the mother. Okay, so that's for 2 marks. So let's quickly look at that. Okay, so basically the offspring inherits an allele for each trait from each parent, right? So from the mother, so just put that M there. So for the mother, they will inherit that gamete. From the father, they have the possibility of either inheriting, sorry, just take that away. Okay, so let's quickly look at that again. So say for the mother, they will definitely only inherit that gamete, because remember the mother is recessive for both conditions. She's homozygous recessive for both conditions. From the father, they have the possibility of inheriting one of the following 4 gametes, because the father is heterozygous for both coat color as well as length of the coat. So they can either inherit this gamete from the father, that one, so they can inherit one of the 4 gametes from the father. So there's basically a 25% chance of inheriting one of these from the father. So in a sense, you can say that the father is the deciding factor for the offspring to have the same phenotype as the mother. Therefore, there's actually only a 25% chance that one of the offspring few of the offspring will inherit the same phenotype as the mother. Okay, hope you guys got that. Okay, so let's move on now to a bit more of genetics type questions. So now we're going to look at a pedigree diagram. So looking at question 2 from November 2017. So what they say over here is color blindness or daltonism is a sex-linked disorder caused by a recessive allele, and as indicated over there. So let's look at this diagram just to make you guys aware, or just hopefully you guys remember this. So your circles indicate your females, and if it's unshaded, they are normal. Your squares indicate your males, and if they are shaded, they're normal. If they're unshaded, they have the condition. Same with the females. If the females are shaded, that means they have the condition. If they're unshaded, they do not have the condition. And as we can see, there are 2 generations represented in this pedigree diagram. Okay. So in question 2.3.1, they say give the phenotype of Senzo with 1 mark. So as you can see, Senzo over there is the male, and he is shaded in, so we can just say he's a colorblind male or male with daltonism. Either option works. Second question, they want to know what is the genotype of Thule. So Thule is over here, she's circle unshaded. So what is her genotype? Okay, so let's quickly just look at this. Okay, so she has XX chromosome, obviously she's female. So because she does not have the condition, she needs to have one dominant allele. However, she would also have a recessive allele because if you look at her offspring, one of them, the son or the daughter, may have inherited the recessive allele with the condition, obviously, from her. Therefore, she would be heterozygous for the condition. Okay, so the genotype of Thule would be heterozygous for the condition. Okay, next question. So describe how Linda inherited color blindness. Okay, so Linda is the female and she is shaded in. So how did she inherit the color blindness? So firstly, Linda would obviously have inherited one recessive allele from the mother and one recessive allele from her father. So, therefore, also referring back to the previous question, that's why the mother truly had to have one recessive allele and one dominant allele. Okay, so back to question 2.3.2, you would get your 2 marks saying Linda inherited one recessive allele from her father and also state the recessive allele and one recessive allele from her mother, and that would be your second mark. Okay, so let's move on to the next question. So continuing with the pedigree diagram question, they now say, explain why there are generally more males than females with color blindness in a population. And that is worth 4 marks. Okay. So males only have one X chromosome. If this chromosome carries the recessive allele, as indicated there, the male will be colorblind. Females have 2 X chromosomes. They need to have two recessive alleles to be affected. A dominant allele on the X chromosome will mask the effect. Okay, so pretty simple over there. So remember, males have an XY chromosome, so they only have one X chromosome. And again, if this chromosome carries the recessive allele, the male will be colorblind. Females have 2 X chromosomes, they need to have 2 recessive alleles to be affected. Right? So obviously, if they had the dominant allele, that would mask the expression of the recessive allele, and then they would not have the condition. So as I said, we have dominant allele will on the other X chromosome will mask the effect of the recessive allele. Okay, so pretty simple over there. So let's move on to the next part of the question. So they say over here, Molly married a normal male. So Molly is over here and she is a normal female and she married a normal male. So that would be basically the square unshaded. Use a genetic cross to show the possible genotypes and phenotypes of their children. Okay, so let's get into that. As I'm going through it, I'll also explain to you guys where you get your marks. So this is usually a 6-mark question. Okay, So first thing what you're going to note is you're first going to say P1, that is your phenotype. Right, so now I need to list the phenotype. So they said Molly is as from the diagram, the Pedigree diagram, you can see that Molly is a normal female. And she is married to a normal male. So now, what is the genotype? So you need to determine that. So let's really look at the genotype of the male. So because males obviously have an XY chromosome, his genotype would be pretty obvious. He would have the dominant allele. Now, because Molly's female, we know that her one allele will be the dominant allele. But what is a second allele? Is it dominant or is it recessive? So Molly would actually have the recessive allele because she inherited the recessive allele from her father, and she would have inherited the dominant allele from the mother, as you guys see from the pedigree diagram. Okay, so now let's start crossing things. So I prefer to do the Punnett square. I just find it a lot neater and a lot more easy to identify what is going on. If you guys prefer the other method, you guys can do that, but I just think the Punnett square is a bit more clearer. So first things first, you're going to list your gametes, and obviously to get your gametes, meiosis had to have occurred. So the gametes from the female, we gonna now, doesn't matter where you exactly put the male and where you exactly put the female. So that is the female's gametes, and the males, we're just gonna list it over here. So now we're gonna have fertilization that would be occurring. So just make a note of that over here. So we're gonna have the dominant alleles that would cross. And then the dominant allele and the Y chromosome, dominant allele and the recessive chromosome, and the dominant allele, sorry, and the recessive allele, and then the dominant recessive allele and the Y chromosome. Okay, so now we have to just basically write down what you have gotten from your Punnett square. So in the F1 generation, the offspring, what phenotypes did we observe? Firstly, from, let's put the point on quickly. So over here we have a normal female. Here we have, so here we have a homozygous normal female. Over here we have a normal heterozygous female. Over here we have a normal male, and over here we have a colorblind male. So that is basically what you write by your phenotype. So you can even write that you have two normal females, one normal male, and one colorblind male. Then what you're going to say is P1 and F1, and then you're going to say meiosis and fertilization. Okay, so let's quickly look at where you get your marks. So your first mark, okay, let's first look at the compulsory marks, the important marks. So you get 1 mark for listing the correct gametes as one of your most important marks. Your second most important mark is for listing the correct genotypes. after fertilization. So that is your two most crucial marks. Then to make up the other marks, you get a mark for listing the phenotype. So that you obviously just get from the question, was provided in the question. The second part you get is for listing the genotypes. And then the other marks that you get is for listing the phenotype. So what you have interpreted from your Punnett square or your genetic cross. That's another mark. And then your other two marks come by simply writing P1 and F1 and Meiosis fertilization. So in total, that's about 7 marks. So you can get 7 marks, but this is a 6-mark question. But make sure you have the 2 compulsory marks because that's what they're looking for. And then whatever else you write, they will mark that accordingly. Okay, so that was pretty simple, straightforward genetic cross type question over there, guys. Okay. So now let's move on to a bit of evolution. So that covers quite a bulk of your paper. So let's just look at how possible ways in which they can ask this type of questions, as well as the different sections under evolution. Okay. So now we're going to look at question 3, that is February, March 2017. Okay. Before we get into genetics, we have one more question I think on. Yeah, so a few more questions to go on genetics. So let's look at question 3 from February, March 2017. So the question reads, hemophilia is a genetic disorder caused by a recessive allele on the X chromosome, right? So a hemophilic female marries a normal male. Explain why all their sons will be hemophiliacs. That is worth 4 marks. Okay, so just to make us understand things a bit better, I'm just going to draw a bit of a I'm going to do a bit of a genetic cross. Okay, so let's say that capital H represents the dominant allele. So let's say again that we're going to say capital H represents the dominant allele, and then small h represents the recessive allele. Okay. So they said a hemophilic male marries a normal hemophilic female marries a normal male. So because she's hemophilic and they say that the disorder is carried on the recessive allele, her genotype for the female would be the following. She marries a normal male, his genotype, he would have dominant allele and chromosome Y. And let's just quickly draw a little Punnett square. I think that's a bit too small. Okay. So again, I'm just going to put a G there for the gametes. And then the female, her genotype, as we know that she is recessive, so that would be her gametes, the male, that would be his 2 gametes over there. So now we're just going to cross and let's see what the genotypes the offspring will have. So let's look at the female. So the one female would be heterozygous for the condition, meaning she will not have the condition, she'll be a normal female. Then we will have another female that again also be heterozygous for the condition. And then the males, so they will inherit the Y chromosome from the father, but because the mother is recessive, they will inherit the recessive allele from the mother and the X chromosome. And then again over here, the same thing. They will inherit the recessive allele from the mother, and they will have the condition and inherit the Y chromosome from the father. Okay, so just to explain things there, as you guys can see. Okay, so an individual inherits one allele from each parent, as we saw in the Punnett square. The Y chromosome was inherited from the father, and the recessive allele was inherited from the mother, as indicated there, since the mother has the two recessive alleles. A son only needs to get one recessive allele to be hemophiliac, since the Y chromosome does carry any allele, does not carry any allele to mask the hemophilia allele. So this diagram basically explains what is written over here. So the son receives the one recessive allele from the mother, and that is the only way in which he can become hemophiliac, and the Y chromosome does not carry any type of allele, therefore the recessive hemophiliac allele can't be masked, so therefore all the sons would have the condition and none of the daughters would have the condition. Okay, so that is pretty simple over there, guys. So I think it's time we're going to move into a little bit of genetics, but I'm just going to take a quick 2-minute break just to quickly have a sip of water. But you guys want to take a quick water break over there, and then we will get back into things. Okay, guys, let's get into it. Okay, so we're going to start off with a bit of evolution now. So we are now going to look at a question from November 2023. So it asks, which one of the following indicates the type of variation for each of the human characteristics given? Okay, so let's do a quick recap. So there are two types of variations, as noted, continuous and discontinuous. So continuous variation, those characteristics where there is a range of intermediate phenotypes, so that would be height. So you have different types that they could be. You get people of different lengths and things. And then you have discontinuous variation, those characteristics that fall into distinct groups, such as blood type. So let's look at the different options. So height would be continuous because people have a range of heights. So option will either be A, C, or D. We're going to eliminate B because height doesn't fall into the same group. Then skin color. With skin color, again, there's a variety of skin colors. Therefore, the option will have to be continuous. Is a variety of skin colors, so skin color would then be continuous, right? So remember we said options are either A, C, or D. So because skin color is continuous, we're going to eliminate the other options. So now, which option are we going to look at? So earlobes. So earlobes, there are two types of earlobes that you could possibly have. So earlobes would fall into the discontinuous group. So that means your answer would actually be B, because again, as when you look at it, height is continuous. There is a range of intermediate phenotypes, different phenotypes for height. People can have different heights, skin color. There's also again a range of different skin colors. Therefore, skin color would also be continuous. And then earlobes would be discontinuous because there are two categories that earlobes can fall into. Earlobes can be lobed or unlobed. Okay, so that's a quite simple, straightforward question. So over here, your answer would be B. Okay, so that's one type of question they can ask under evolution. Let's look at another type that they like to ask. But let's quickly just look at a little bit of theory with regards to this question. Okay, so phylogenetic tree. So let's have a look over here. So there are different ways in which they can show phylogenetic trees. Okay, so the first one over here, look at this one. So you have 5 different types of species, and this phylogenetic tree is read from left to right. So you have your ancestors, the older species, and you're going to your new present day species on the side by A, B, C, D. Then let's just look at a closer up, or let's start here with the ancestors. So over here, the black dot here represents the most recent common ancestor of the following species, A to E. But now if you look closer over here, the most recent ancestor or most recent common ancestor of A and B is located by that dot over here. The same would apply to C and D. Their most recent common ancestor would be over here. Okay. So also again, phylogeneity is basically the representation of the evolutionary relationships between species. So another way in which they can show the diagram is over here. So firstly, you have your branching event that happens here at once. So branching event can also, what that also is basically a speciation event where a new species began to develop. So the common ancestor for all the species in this phylogeneity tree is the one at number 1. So let's look a bit closer at the different species. So A, B, and C, they have a common ancestor at 2. And then a speciation event occurred, and then with B, we have B and C over there, then B and C, now the speciation event occurred, and then number 3 over here, that would be the common ancestor of B and C. So B and C are more closely related to each other than to A, but A, B, and C are more closely related to each other than D and F, and A, B, and C are more closely related because they have a common ancestor at 2. Then in a case where one of these lines end, that means an extinction of that specific species. Okay. So let's move on, guys. So we're going to look at a question that has a bit to do with your phylogenetic days. So November 2017. So we have 3 related species, X, Y, and Z, and they share a common ancestor. So X, Y, and Z share a common ancestor. Species Y and Z share the most common recent ancestor. The species Y and the species Y and Z share the most recent common ancestor. Which phylogeneity most accurately represents their evolutionary relationship? Okay, so let's look at it. So now I think what we can do with this question is it's best just look at in which of the 4 examples, where is Y and Z sharing the most common recent ancestor? So let's quickly look at A. So over here, Y and Z do not share the most recent common ancestor because X and Y are more closely related. In B, Again, over here, it appears that X and Z seem to be more closely related. So A and B is totally out. With C, here we have, as the question wanted, Y and Z are the more closely related species and they share the most common ancestor. All right, so C is possibly the option. It's possibly the answer. Then in D, we have Z and X sharing the most common ancestor, not Z and Y. Therefore, the answer had to be C. This over here clearly indicates that Y and Z share the most common ancestor. So this is just a process of elimination type of question, but it is also a multiple choice question, so it's about worth 1 mark. Okay. So guys, let's move on to another type of phylogenetic tree. So let's get to it. Okay. So this is from Gauteng, September 2021. The phylogenetic tree below shows the evolutionary relationships between some species. So again, read from left to right. Okay. So let's look at the first question. What do they want to know? If the letter of the most recent common ancestor of all the species in the diagram. Sorry. So give the letter of the most recent common ancestor of all the species in the diagram from the theory that we went over a little bit not too long ago. It would be A. So A represents the most recent common ancestor of all species. Right? So just take note of the wording of they ask for the most recent common ancestor of all species. Okay, next question. When did the chimpanzees and modern humans diverge or separate from the common ancestor? So when did the chimpanzees and the modern humans diverge from the most recent common ancestor? C. So now they want basically the time period. So let's look at your timeline over here. So this looks like it's about on 6. So it's 5, 6, 7, 8, 9, 10. So there's a 1 million gap between each space. So the answer would be that chimpanzees and modern humans diverged or separated from the common ancestor C about 6 million years ago. If you want to say maybe like, I don't know, 6.5 or 5.5, there's usually like a range that they give you. So if you're within that certain range, they will give you the mark. Okay. Now they ask you to state 3 characteristics of the upper limb that humans share with other primates. So this is just from your studying knowledge. So state the characteristics of the upper limb that humans share with other primates. Firstly, they both have freely rotating arms. They have long upper arms. They have rotation around elbow joints. They have bare fingertips or nails instead of claws, and they both have the opposable thumb. Okay, that's pretty straightforward over there. Okay, so next question. Question 3 from Gauteng Province 2020. So the diagram below shows evolution over a period of time and is characteristic of punctuated equilibrium. So let's look at what they're asking us before we start interpreting the diagram. So they would like to know, use the labels A and B to explain punctuated equilibrium. So they're saying to use the label. So in your answer, that's worth 5 marks, you need to explain what the labels A and B mean, what they represent. If you don't list that, you would not get the marks. Okay, so let's look at it. So let's quickly just define what is punctuated equilibrium. It explains the speed at which evolution takes place, right? Okay, so the first statement, punctuated equilibrium, explains the speed at which evolution takes place. That is 1 mark. So now we can explain the different points or the labels on the diagram as they wanted. So A shows a long period of time where species do not change or change where very little change occurs. And this is known as equilibrium. So you get another mark, or one of the compulsory marks, as they say, would be A shows a long period of time, as indicated on the diagram. So that is 1 mark, and that's a compulsory mark. Second mark would be where you're saying that A is where species do not change, or there's very little change. That would be your second mark, and this is known as equilibrium, a period of no change. That would be your fourth mark. Then this alternates with the short period of time at B. So as you can see on the diagram, B is a short period of time where rapid change occurs through natural selection, during which species may form in a short period of time. So to get your fifth mark, you're going to explain what's happening at B, short period of time. That's 1 mark. Where rapid change occurs, that would be your second mark. And as a result of all of this, or as a result of the rapid change at B, this would be where your new species may form. Okay, so that would be how you would actually get 7 out of 5 marks. But obviously, again, when you listing all your points, make sure that you list the main points that they're asking for, labels A and B, and then whatever else follows, make sure it's in chronological order, and that is how you will get all your marks. Okay. Right, guys, so moving on to the next type of question. Question 3, this is Gauteng again, 2020. Okay. So, a lake was once full of water and a population of fish was able to move freely throughout the lake. A severe drought caused the level of water in the lake to drop, such that the population of fish became separated, as shown in the diagram below. Many years later, it was discovered that speciation had occurred. So, let's quickly look at what, just quickly define what is speciation. So, speciation is basically as a result of natural selection taking place over a period of time. The characteristics of the organisms may change to such an extent that they cannot reproduce with the original members of the population. So let's say the big pond over here is population A, or the big lake is population A, the small lake is population B. So natural selection took place, and the characteristics of population A and the characteristics of population B changed, so that if they ever met or interacted, they would they cannot reproduce with the original members of the population, which is population A, and they would not be able to produce basically fertile offspring. And this is because each of these two populations, A and population B, they have now become a new species, meaning speciation had occurred. Okay. So let's look at the questions relating to this diagram over here. Okay, so it says describe the type of speciation illustrated in the diagram. So that's worth 6 marks. So let's look at it. So again, it's really just a recap. A lake was once full of water and a population of fish was able to move freely. So this population was separated because the water levels dropped. So we have now over here population A, population B. So what type of speciation occurred? That would be geographical isolation. So speciation occurred through geographical isolation. And this occurred on the rocky island, and the geographical barriers separated the fish into 2 populations as indicated. So your first mark would be to describe the type of speciation that occurred. And by saying speciation through geographical isolation, that is 1 mark. Where did this speciation occur? On the rocky island or on the land? That would be your second mark. And the geographical barrier separated the fish into 2 populations. That would be your third mark. Okay. Then, because the 2 populations are separate, there is now no gene flow between the 2 populations, right? That would be your fourth mark. And with different environmental conditions or different selection pressures happening on each side, natural selection occurs independently in each population. So by saying different affected by different environmental conditions or selection pressures, that would be your fifth mark. And natural selection also occurs independently, that would be your 6th mark. Okay. Then, as a result of natural selection occurring and environmental and the effects of environmental conditions, each population becomes phenotypically, they look different, and genotypically the genetics are different from each other, which prevented them from interbreeding, leading to the formation of new species. So by saying they're genotypically and phenotypically different, that is your 7th mark. And by also saying they can't interbreed, leading to the formation of new species, that would be your 8th mark. So you can get 8 marks for basically a 6-mark question. Okay. So pretty simple over there, guys. So basically, this is just, it's basically a template for speciation. So you just have to, in a sense, memorize the general order of how speciation occurs. And then when you have a question that is asking you to be specific to that question, you just basically have to fill in the gaps with what the question is providing. They either might ask it in like this, or they might just ask you to just explain speciation in general. So you can go either way. Okay. So let's move on. So continuing with the speciation question. So basically, the question reads, different species of fish breed at different times of the year. Describe how this reproductive isolation mechanism helps keep species separate. So again, there are, remember, different reproductive isolation mechanisms. Just to recap, there's breeding at different times of the year, as we will now discuss. There is species-specific courtship behavior. There is adaptation, different pollinators in plants, as well as infertile offspring. So those are the 4 main reproductive isolation mechanisms. But now in this question, they're asking us just to focus on breeding at different times of the year. How does this affect the species, and how does it keep the different species separate? So firstly, the male and female fish of a particular species only become sexually active during certain times of the year or certain season, right? So other species may not be sexually active or have different breeding seasons. Therefore, they are unable to mate with other species of fish, thereby keeping the species separate. Okay, so that is your 3 marks. So male fish of a particular species only become sexually active during certain times of the year, so that is about 1 to 2 marks. And then the other mark will come from other species may not be sexually active or have different breeding seasons, therefore they are unable to mate with other species of fish, thereby keeping the species separate. Okay, perfect. So now we're going to move on to our last set of questions. Okay. So now we're going to look at question 3 from November 2018. Okay. So it says over here, the diagram below shows the upper jaws of some fossils. These diagrams are drawn to scale. So chimpanzee, Australopithecus afarensis, and Homo sapiens. So with this, we need to analyze the diagram because the following questions are all going to be relating and referring to the diagram. Okay. So question 1. Describe one visible difference between the jaw of the chimpanzee and that of the Homo sapiens, which show trends in human evolution. So describe one visible difference, and that is worth 2 marks. So there's a one difference, and it has to be a visible difference. So what you see here, it can't be anything else. You can't be speaking about the cranium or bipedalism, but here they're just referring to the jaw and the mouth. Okay, so let's look at the possible things that you could say for this question. So the jaw is large in the chimpanzee and is more rounded in the Homo sapiens. You could also say the jaw palate is rectangular in the chimpanzee and rounded in the Homo sapiens. So again, it's worth 2 marks because you're saying what's happening in the chimpanzee and you're saying what is happening in the Homo sapiens. So you get 1 mark for mentioning each species. You could also say large spaces between the teeth in the chimpanzee. There are large spaces between teeth in the chimpanzee and small or no spaces in the Homo sapiens. Then you can also say that there are large canines or teeth in the chimpanzee and small canines or teeth in the Homo sapiens. So you can see over here, we're going to be referring to teeth. You can refer to the spaces by the teeth. You can refer to the jaw as a whole or the palate. So there are 4 possible visible differences that you can make note of and so on. Okay. So next question. Based on the difference in dentition, the teeth, what conclusion can be made about the change in diet from Australopithecus afarensis to Homo sapiens? That is worth 2 marks. So Australopithecus, they have more also quite big teeth. They have a bit of canines, whereas Homo sapiens have much smaller teeth and a very small canines. So what differences? How did this change teeth difference also change the diet? So the diet change from eating raw food in Australopithecus to eating a diet of more cooked food in Homo sapiens. So that is your 2 marks. Okay, so pretty simple over there. So question 3, continuing with this question. So they say where Australopithecus may be described as a transitional species between the chimpanzee and the Homo sapiens. So this define a transitional species. So define another question. What does define mean? Just gives a clear meaning. What is, they just want the definition. So don't list any traits that the Australopithecus has of the chimpanzee and the Homo sapiens. That could be another question. So just define, give a clear definition. So what do we say for a transitional species? So a transitional species shows intermediate characteristics between 2 generations. You can either say that, or you can say it has characteristics. Transitional species has characteristics common to both the ancestor species and the species that follows. So you have 1 or 2 ways of saying that question, and it's only worth 1 mark. Okay, question B. It says use one visible feature of the jaw to explain why Australopithecus afarensis may be described as a transitional species. So one visible difference, so what you see from the jaw. So you could say that the jaw of Australopithecus is smaller than that of the chimpanzee, but larger than that of the Homo sapiens. So again, you're listing 2 features. So you're listing the features of the chimpanzee, but you're also listing how it's different from the feature of the Homo sapiens. So that's how you get your 2 marks. So we have to make a comparison between chimpanzee and Homo sapiens. What you could also say is that the canines are smaller of the Australopithecus are smaller than that of the chimpanzee, but are much larger than that of the Homo sapiens. Or you could say that the jaw or palate is more rounded than that of the chimpanzee, but less rounded than that of the Homo sapiens. Okay, so that is one question on your another question on your evolution. Okay, so let's look at our last question over here. So this is question 3 from Gauteng Province 2020. So the map below illustrates a possible theory on how modern humans migrated to all parts of the world. Okay. So the first question, name the hypothesis that supports this theory. So how modern humans migrated to all parts of the world. That would be referring to the out of Africa hypothesis. So that hypothesis just states that modern humans originated in Africa and migrated to other parts of the world. That's just 1 mark. Quite simple. And then next question, describe the genetic evidence that supports the hypothesis in question 3.5.1. So they only want to know the genetic evidence. Okay. So what you can say is that the mitochondrial DNA is only inherited through the maternal line and is passed from mother to offspring. Okay. So you get your 1 mark by saying mitochondrial DNA is only inherited through the maternal line. That is 1 mark and is passed from mother to offspring. 2nd mark. So the analysis of the DNA shows that Africa was the location of the oldest female ancestor. So that would be your 2nd mark. And the oldest female ancestor is mitochondrial Eve, and then all humans descended from her. That would be your 4th mark. Okay. So remember, only genetic evidence was asked for, no other evidence. Okay. Then the next question. They're asked to describe the fossil evidence of hominids found in Africa that supports the migration of modern humans as shown in the map. Okay. So first things first, fossils of Ardipithecus were found only in Africa. So that's one mark. Second mark would be fossils of Australopithecus were found only in Africa. And then third mark would be fossils of Homo habilis were found in Africa, only in Africa. And then your fourth mark would be the oldest fossils, Homo erectus and Homo sapiens, were found in Africa. So that would be your 4 marks. Other statements that you can make is that the younger fossils of Homo erectus were found in other parts of the world, and the younger fossils of Homo sapiens are also found in other parts of the world. Okay, so that is actually the last question for us, guys. I would like to say thank you guys for joining us for our final session for our grade 12 masterclasses. I definitely know that all the maths and physics and life science sessions definitely helped you guys out quite a bit. I hope you guys are checking out our YouTube channel and just liking and subscribing all our videos. And I definitely know that it's helping you guys. Thank you guys for joining us throughout this masterclasses as well as our spring schools. And I wish you guys all the best with your studies. I know you guys have done your best in all your subjects and you're going to get super high marks in all your maths and physics and life science papers because those are the most important topics over there, guys. So all the best, guys. Till next time, I know you guys are going to do well with your results. See you guys.